Hello everyone, this is MEENU AGRAWAL. Now continuing with this human reproduction, we are moving to part 3, and in the previous section we have dealt with female reproduction, isn't it? Where we have seen their structures, oogenesis, and we have even dealt with ovarian cycle, uterine cycle, all those features, hormonal impact, all those things we have discussed. So now moving on, now after this what is going to happen is now we will go to start with fertilization events. So now if you look into the figure, now if fertilization doesn't happen, what happens? We have seen in menstrual cycle. Now what happens on fertilization? Let's move on with that. Okay, so this is the figure which is showing you that now when the sperm comes, like this is the figure which is dealing with the fertilization of a sea urchin. Okay, and in case of sea urchin there is an external fertilization which is most commonly easily available, and therefore a lot of research work has been done on sea urchin. So why I have taken this diagram, I will tell you, and it's also related to humans also, I will tell you. See, what is happening here, you can see here that as the sperm comes in the vicinity, as you can see, this is written as what? Jelly coat. Now they have got an external fertilization and their egg is in that jelly coat. Okay, and when the sperm comes in contact with that jelly coat, what it does, it releases this acrosomal enzymes which are hydrolases. Okay, they now they start digesting and they form what? Acrosomal processes, that is your actin filament, and they release certain proteins. Okay, you can see that they release certain proteins that binds the receptors present on the membrane. Okay, and that opens a gate for them, for the sperm nucleus to move into the egg. Okay, as the two membrane fuses, what happens, that the sodium ions open, and there is a movement of sodium ions inside the egg, and as a result, you will read about this depolarization. Polarization takes place, and in this depolarization is an what is happening, the ions are involved and they are changing the potential of the membrane. Okay, you have read about the potential in physics, same thing is here, that the potential of the membrane changes, it becomes depolarized, and as a result they prevent now any other in the vicinity, if any other sperm is there, it is not able to bind to the egg. And why is it that it is electrical in nature? First of all, you understand that it is what? Electrical in nature, and it is very fast process. Okay, this is called as fast block polyspermy. Fast block polyspermy. And fast blocks polyspermy, and then later on, to further prevent uh, that no other, now because in ocean and all, what is happening ki there mostly external fertilization is there, and other species sperms are also there, so not allowing them to fuse with, what they do, first they do depolarization, that is fast blocks polyspermy, and then they do what, that this binding with the receptor, it creates a kind of a signal. Okay, the signal causes the release of what? The signal, there is a signal transduction. Okay, so via signal transduction, what is happening, a kind of a signal is given, you will read about them more when you will read about hormones and all. Then what will happen, it will cause the release of calcium ions, it will release. Okay, these calcium ions will cause now the bursting of these cortical granules which you are seeing here. Okay, so these cortical granules, what they will do, they will burst, and what will happen, they will clip off. What will these cortical granules will do, they will clip off these receptors. As you can see there, they are clipping off the receptors. Okay, so you can see that they will clip off the receptors, and therefore no more, if any sperm is bound also to these receptors, will be clipped off. Okay, so they and they fill that space between your what, this is your what, vitelline membrane, this is your what, vitelline membrane. Okay, uh, let me see the cursor. So this is your what, now vitelline membrane, and you know that vitelline membrane is formed by the extracellular material of the egg, and this vitelline membrane then raises up. Okay, it raises up, and this layer which is formed, now the cortical granules, and this forms your what, it forms your fertilization membrane, so which doesn't allow any sperms to enter into it. Okay, so this is how this blockage is there, and this blockage by calcium, this release of cortical granule, this is your slow uh, block, this is called as slow block polyspermy. And remember one thing, that you see this both in the case of sea urchin, but in case of humans we only see what, we only see slow block polyspermy, we don't see fast block polyspermy in humans. Why? Because in humans, you see, when the egg enters into the female genital tract, then what is happening, it is uh, gaining this ability now to, to become more active, and it becomes more able to move. Okay, and rather you can say the capacity to fertilize, the egg, it gains in the female reproductive tract, and that process is called as capacitation. Okay, so lot many changes takes place there, and then what happens, then this sperm, and it's not an external fertilization, therefore immediately they don't have to block. So what they do when they till here the process is same, but they doesn't takes a depolarization. What happens, they signal due through signal transaction, what is happening, calcium will be released, and they will cause the bursting of the cortical granules, which will clip off the receptors. Okay, and will prevent polyspermy. Okay, so now let's move on and see what happens when the, remember that this fertilization event which is taking place, which is also causing one thing major is there, a lot of it is in causing the metabolic changes in the egg, and making the egg more metabolically active, and it has been seen that they start forming more. So as the sperms enter the egg, what is happening? Now egg, there are certain changes taking place in the egg. The egg is becoming what? Metabolically active. It is becoming what? Metabolically active. Like certain changes can be seen, more proteins are being formed, more proteins will be formed. Okay, and what else will take place, your respiratory enzymes will become more active, so respiration will start, all these processes will start taking place. Okay, now what will happen, now after the zygote is formed, your zygote will then start dividing. In case of humans, the first division takes place at around your 16, around 12 to 32 hours after that. Like in case of sea urchin, it takes place very early, it's around 90 minutes. So with different organism it varies. Now what do you see here, that as now there is a term which has been used which is called cleavage. Okay, why we are calling it as cleavage? Because the cell is dividing, but what is happening, there is just the cytoplasmic division हो रहा है, and what is happening, cytoplasm is dividing and your DNA synthesis is taking place. Okay, and what we are finding, that the growth of the cell is not taking place. Okay, So what do we find? That no proteins and all are not being formed. When the growth will happen, then these things will happen. So what is happening? Basically, it's a kind of a redistribution of that cytoplasmic material which is taking place. And these cells are called as what? What do we call them? We call them as blastomeres. What do we call them? We call them as blastomeres. Okay. And at the morula, what do you find? This is the stage where the cells are at what? 8 to 16 cell stage. Okay, they have 18 to 16 cells in them, and after morula comes your, this stage is called as blastula. Okay, so at this stage of blastula, what is happening? You can see that the implantation is taking place. Your implantation of your embryo takes place at this stage of blastula. Now let's look at this diagram more clearly. Now you can see here in this diagram where I'm showing you, this is your oocyte. Now it has divided. Now by the fusion of the sperm, it has formed zygote, and then it has divided into corolla, which is around 8 to 16, 16 cell stage. Okay, now what do we find further? That this is your what? Your blastula. Okay, which is consisting of an outer layer of cell which is called as what? What do we call these cells as? We call them as trophoblast cells. Okay, and this is called as what? These blue ones, can you see? They are called as your inner cell mass. Okay, which will trophoblast cells will give rise to your what? It will give rise to your extra embryonic membrane. It will give rise to your extra embryonic membrane. Okay, and your inner cell mass, it will give rise to what? It will give rise to your embryo. First of all, these things must be very clear, crisp and clear to you. Now moving on, what do you see? There is a term which I am trying to highlight you, the difference between the two terms. What are that? That is your totipotent cells and pluripotent cells. Remember one thing, that they are both are stem cells, and both are your what kind of cells? Already we have talked about it earlier in other videos. They are undifferentiated. They are undifferentiated cells. Then what is the difference? The difference is that your pluripotent cells can differentiate and give rise to other cells of your body, and totipotent cells can also differentiate and give rise to other cells of your body, plus it will also give rise to the cells of placenta. It will give rise to the cells of the placenta, which your pluripotent cells cannot do. Okay, now at this moment I will also try to explain you about, you must have heard the term monozygotic twins. Now if there is a single zygote, okay, zygote is there, and that zygote divides, okay, that zygote divides and it forms two cells, and somehow these cells get separated. Okay, when they get separated and they grow as an individual embryo, and then we get what? Two individuals we will get in the womb. Two or children will be there, baby will be born, and they will be what? They will be monozygotic. Look at the word mono. So they have been formed by the same cell, same zygote. So genetically they are identical, isn't it? So we call them as what? Identical twins. And I hope all of you must have seen such cases. So they are identical in all the respect. Okay, and you must have also heard about, I must also explain about the other one also to you people, that is your dizygotic twins. uh, in this case what is happening, okay, this case is like any no siblings will be born, but in this case what is happening is that generally you know ki one ovary ovulates at a time. Okay, if right side is ovulating in 1 month, and the other month the other ovary will ovulate, but sometimes what happens, both the ovary ovulates together. And I have told you there are a lot of environmental factors which operates on this menstrual cycle on female's body, their psychological factors, so many factors are there. Okay, so what do you find that this both the ovaries releases the egg and both the eggs get fertilized. Okay, therefore in case of fraternal twins, what do we find? We find that in case of your fraternal twins, what do you find? That their sexes can be even different. Okay, their sexes can be even different, but this is not the case with monozygotic twins. The sex will be same, but here the sexes can be even different, and they are non identical. They don't look similar, in any respect they don't look similar. Now moving on. Now you can see here that an implantation has started, and what is taking place is here is that now you can see here, now they are forming. We will see further how the complication is building up. Now you can see this here is your embryonic disc. Okay, which will further now it will give rise to all the three layers of the cells, that is your ectoderm and mesoderm and your endoderm. Okay, now your ectoderm, as you know, will give rise to your nervous system, skin. Mesoderm will give rise to your muscles. Endoderm will give rise to your gut system. Okay, so now as you move on, what do we find? That these are the three germ layers, and at the 20 days, what do you find here is that now the embryonic membrane can easily be now clearly be distinguished. Now you can see here, now there are the three embryonic membrane. Okay, that is amnion. Okay, now I'm going to talk about them in detail. So amnion, chorion, allantois, and your yolk sac. So here what do you see? This is your yolk sac. Okay, this is your yolk sac. Okay, now at the 20 day you can see the embryo has also grown quite a lot. Okay, now just let me show you that the embryo has grown quite a lot, and you can see here that the formation of head has also started. Okay, now what is happening, this yolk sac, see, in case of other organisms like bird and reptiles, yes, they have a role that they're laden with food, with nutrient, but in case of mammals they don't have this role, and they're quite diminished in a way. Just they are the site for the first blood cells, they are the site for, okay, site for first blood cells to be formed. Okay, and they are the site for they also contribute, they are the site where your spermatogonia, the spermatogonial cells have come, and also they are the site where your oogonial cells have come. Huh? Oogonial cells have come. Okay, so they are the site for that also, and later on they become what? They part of your umbilical cord. This is what, let me show you here. This is what is your umbilical cord is being formed, although it will grow more. So you can see this is an umbilical cord, and it will become a part of your umbilical cord. Now if you look at this amnion, amnion is what? It's forming what? It is a kind of a fluid filled sac. Now if you look at your, now wherever the external fertilization is taking place, in them what do we find? What do we find? We find that their water is already a medium for them, and since these from reptiles, if you look at, to birds if you look at, they are completely terrestrial, and the how to provide that liquid environment to the developing embryo, so it was provided by this therefore these three reptiles, mammals and birds are called amniotes. They have this amniotic sac which is filled with a fluid. Okay, and this fluid has got lot of role, as you can see here in this diagram. Okay, so let's look at here. You can see here now this outer now is what, it's your, this is your amniotic fluid, isn't it? So this is your amniotic sac, okay, and which is now containing, which is filled with fluid, okay. Now it helps the baby to float in that fluid. Even on growth, it forms kidney, it urinates in that fluid, okay. So with that also the cells of that baby comes into it, which is basically very important. Now clinically it is very important, as it helps it helps the um, uh, to know about various diseases, genetical disorders, and that technique we are going to read later on, and that is called as amniocentesis, okay. So this is there, and also you know that this rupture of amnion is a sign for what, for your labor pain, or you can say is a sign for that that the baby is going to be born, then labor is beginning, okay, or parturition is going to take place. Now the next layer is your chorion, which I have told you, as the name is given to the trophoblast cells, and it will develop further and will become what, interdigitated with your uterus and form small projections called as chorionic villi, the fetal blood vessels, okay, and it will become what, the fetal blood vessels, and begin to grow into endometrium. So you can see here what they are forming. See, you can see here these are forming your what, chorionic villi. These are your chorionic villi, okay, and inside it what is happening, the fetal blood vessels are growing, okay. I will show you the placenta diagram much more clear than this, okay. Now what do you see, contains the fluid blood vessels that becomes the fetal part of the placenta, okay. And your alien toys has got no, not major role, but just helps in taking out the waste, and they become a part of your what, they become the part of your umbilical cord. This is the cord by which the child is connected with the mother. So you can see here that this is your what, this is your umbilical cord, okay. So now you can see this diagram which is showing you really the structure of the placenta, okay. Now what do you see here, what do you see here, you can see here now, now remember one thing, that here now the arteries will take the blood from the fetus to the placenta, to the and from placenta your veins will now bring the blood, okay. So remember which will contain more oxygen, and like that of questions if comes, so you have to remember which is taking the blood. And you know the role of the placenta. Placenta is involved in what, in exchange of gases, in exchange of now taking nutrients from the mother's blood to the baby's blood, and it is also responsible for removal of waste from the fetal blood to the mother's blood, okay. So what do you see here, basically there is no direct contact between the mother's blood and the child's blood, embryonic blood. Why? You can see here, see, these are what, these are the your maternal arteriole, okay, and they are now opening into your what, maternal sinuses, okay. So this region is called as what, sinus. Let me show you this region, okay. You can see here, okay, and these are your chorionic villi, in which you can see here in this chorionic villi your now your embryonic arteries and veins are coming into it, and these back portion now blood, these are open spaces, okay, and these are sinuses, okay, in which your this blood is coming and opening. So they are not in direct contact, okay, so they are not in direct contact with each other, okay. So which you can see here very clearly, the diagram is illustrating this, fine. So this is there, okay. And umbilical cord is the main cord which connects the mother and the baby. And I have a question for you people, like if like in case of dogs and all, you people see they give rise to 4 or 5 puppies. What do you think, that they have got that many umbilical cord, or a single umbilical cord with all are connected? Think and try to answer it. If possible, you can write in the comment box also. Okay, so and when the baby is born, then what is happening? We will look at how they cut the umbilical cord also. Okay, so